Now we're going to take a look at a way cells are able to move really large particles in and out. When you learned about the cell membrane, you learned about this phospholipid bilayer, the two layers here, phosphates once again, lipids in the middle. And one of the things to keep in mind is that it's almost a bit more like a blanket. It can fluid, it can flow back and forth, it can reshape itself, almost even like a plastic bag that you could tie a knot in. And that property, its flexibility, is part of what makes it able to move really large and small things in and out of the cell. We already saw how cells can use carrier proteins to move larger molecules in and out. But we're going to look at how they can move really big ones in and out that sometimes get packaged by the Golgi apparatus. So our first example is you've got a solid food particle here, and this food particle needs to go into the cell. It'll approach the cell membrane here. Part of the plasma membrane will start to scoop around it, eventually totally surrounding it and engulfing it, and then the little pod that's left over is free to move into the cell. This is called phagocytosis, and it is how cells can get large pieces of food into a cell. A much similar process is called pinocytosis, and instead of food going into the cell, you can see liquids and maybe a few little minerals or dissolved ions can get sucked into the cell too. Once again, the cell membrane circulates around it until it pinches it off, just like that, and it's able to move freely into the cell into a vesicle where later those nutrients can be received. These two processes collectively together are called endocytosis, endo meaning going into a cell. So you have endocytosis, pinocytosis, which is for fluids, and you have endocytosis and phagocytosis, which is for food. The opposite of these two processes, of course, would be called exocytosis, when things leave the cells. We know the Golgi apparatus has the ability to package up these materials into vesicles, whether it's a solid or liquid, and these vesicles will head to the surface, and much like in the endocytosis process, but in reverse, as this approaches the surface and it comes in contact with the surface, then this cell membrane will break off along the outside edge and allow those materials to flow out. That is exocytosis. So if we look at the big picture of how this works in cells, we've got our little ribosomes here. We've got our endoplasmic reticulum, the ER there. There's some smooth ER here, which is going to be more for producing lipids. The rough ER with the ribosomes obviously is for proteins. These will get packaged into vesicles and go to the Golgi apparatus, which will further package them into vesicles that can go out of the cell and through exocytosis leave the cell membrane. And that's the way some of these nutrients can get out of the cells that our cells might produce. So we've got a process going ribosomes and ER, which once again that's for the proteins, and some of the ERs for the lipids as well, to a vesicle which goes to the Golgi body, which will package it and ship it out as another vesicle which leaves the cell membrane through exocytosis.